Welcome to The Explainer. Today, we're diving into a really powerful technique that's totally changed the game for genetic research. Think of it like a high-powered searchlight that can find the tiniest, most important typos hidden somewhere inside our massive genetic code. So, to really wrap our heads around the scale of this, let's start with an analogy. Imagine your job is to find one single critical typo in a library filled with 1,000 books. That's pretty much what scientists are up against when they're hunting for the genetic causes of a disease. It's a huge task.
Okay, but what if I told you that you had a secret weapon? What if you knew that almost all the important typos were packed into just 1% of all the pages in that whole library? You wouldn't read every single page, right? Of course not. You'd go straight to that 1% and focus all your energy right where it counts.
And that's pretty much the secret behind the exome. You see, while the exome only makes up about 1 or 2% of our entire genome, it's packed with the good stuff. It holds an estimated 85% of all the mutations that are known to cause diseases. It's the most valuable, action-packed part of our DNA.
This is where a brilliant technique called Whole Exome Sequencing, or WES, comes into play. It's designed to do exactly what we were just talking about, read that crucial 1% of the genome. It gives researchers a focused, powerful, and efficient way to look into our biology. So let's break down how it all works.
First, let's really dig into the problem that WES was built to solve. We're talking about finding a genetic needle in a truly gigantic haystack.
I mean, our genome is just enormous. We're talking 3 billion DNA bases. Trying to search that whole thing for one tiny mutation linked to a disease, well, it can be incredibly slow, super expensive, and you end up with an amount of data that's just overwhelming to analyze. It's like trying to scan and cross-reference every single word in that entire 1,000-book library. So instead of that brute force approach of reading every single letter in every single book, WES gives us a much smarter, more targeted way to get the job done.
You know, this quote from our source material really hits the nail on the head. WES hones in on the areas with the greatest genetic value, and by doing that, it lets researchers massively increase what's called sequencing depth, basically how many times each little piece of DNA gets read. This is absolutely critical for accuracy, and you get to do it all while keeping costs from spiraling out of control.
So the big question is, how does this targeted method actually stack up against sequencing the entire genome, a technique we call Whole Genome Sequencing, or WGS?
Well, at a glance, the advantages for a lot of research projects are crystal clear. WES is way less expensive, it's faster to get results, and it produces a much smaller, more manageable set of data for your bioinformatics team to actually work with.
And what's really cool about this chart is how it positions WES as this perfect sweet spot. Look, if you need to see absolutely everything, you go with WGS. If you already have a handful of genes you're suspicious of, you'd use a targeted panel. But WES, it's the ideal choice for discovery, for finding new things in those crucial coding regions, but without the massive complexity and costs of the full genome.
So here's the bottom line. WES gives you this incredible combination of efficiency and accuracy. You get really deep coverage, often over 120 times, which means each bit of DNA is read over and over and over again. That gives you super high confidence in your results, makes the data really reliable, and lets you process a whole lot more samples more effectively.
Okay, so we've covered the why. Now let's jump into the how. Let's see how this all works in practice, from taking a biological sample all the way to getting actionable data. So while it's super technical in the lab, the whole workflow really boils down to 4 main steps. First, you isolate DNA and break it up. Second, and this is the key step, is what's called exome capture. Scientists use these tiny molecular hooks that are designed to go in and fish out only the protein-coding bits of DNA. Third, that captured DNA gets sequenced. And finally, powerful computer analysis turns all that raw data into a clean list of genetic variations.
Now, that workflow might sound pretty straightforward, but each one of those steps is incredibly complex. And the truth is, the quality of your final data, and really the success of your entire research project, depends completely on how well every single part of that process is executed.
And this right here is where having an expert partner becomes an absolute game changer for researchers. Getting clean, reliable data that's ready to analyze is the difference between a successful project and one that's just bogged down by false leads and frustrating reruns. This is exactly where a specialized service like CD Genomics can make a massive difference. For instance, by using optimized capture kits, they make sure you get better, more even coverage across the whole exome. Their standardized quality control means you have less uncertainty. The whole goal is just to remove those common headaches for researchers and deliver data that's ready to go, making your entire downstream analysis that much smoother.
So they give you everything you need to just hit the ground running. You get the raw data files, the aligned data files, and most importantly, the variant call files, which is the actual list of genetic differences they found. But on top of that, you get annotated tables to help you make sense of it all and a full project report. This ensures your data isn't just generated, it's actually ready for you to interpret. So at the end of the day, whole exome sequencing is just an incredibly powerful and focused tool for making scientific discoveries. It helps researchers find those meaningful signals in all the noise, and that speeds up our understanding of everything from rare diseases to cancer. It really just leaves us with one final thought. The answers are waiting in the data. The question is, are you ready to find them?